Thank you so much for allowing us to present today. It's great. My name is Arlene Nugent. I'm a researcher at AbbVie. I work in their Global Medical Affairs Department, and I'm actually tag teaming this presentation with my colleague Anna, who's sitting at the front, who is actually one of our medical monitors, our lead medical monitor on the program. And we also have Dan. Dan is over here. He's our global development lead. So the three of us are here together. So I think I'll start. And what we wanted to talk a little bit about was our program, but also the context of how do we do immune-mediated viral control, often antiretroviral therapy. And so some of the things that we're going to talk a little bit about today are things that you already heard early this morning. So I'll just touch upon them again briefly. And then really what we want is we want to hear Anna talk about what we've been doing in the clinic and what we've been seeing. So we'll go to the next slide. This is just a disclaimer slide, and just to share with all of you that what we are studying are investigational agents, so they have not been approved yet by any regulatory authority. And so we no way recommend or imply that we use products for unapproved uses until they're studied. Okay, so we'll go to the next slide. Okay, so this is a very similar slide to maybe what you saw earlier today, but really we're very lucky in the era that we're at is that we've had a lot of changes in the trajectory of HIV treatment over the years. We have great antiretroviral therapies today. And even in the next couple of years, we're going to see even longer acting antiretroviral therapies, whether they're orals or injectables. But the big thing is, as you've heard, is that we can still not get to the viral reservoir. So how do we get to the gold star, the North Star, which is cure, right? Which is where we can actually get rid of all of the virus in all of the tissues, or even nullify it so that we're basically causing it to sleep forever, so it can't do anything in the body. And so one of the ways is sort of this middle part where you see 2030 is we can maybe use regimens where you do sort of like a finite treatment period and get to long-term viral control, viral suppression, without having to take antiretroviral therapy for a long period of time. And what I mean by long-term is, at least 1 year, but hopefully 2 years, and even in the future, longer than that. So we'll go to the next slide. So there's a lot of considerations, though, that we have to have when we're doing this kind of research where we're taking people off their antiretroviral therapy. Some of the factors is that the first thing is that we want to make sure that we're not, when people are off of their ARTs, we're not further damaging anything or further increasing inflammation as they're off of their antiretroviral therapies. The second thing is that we can learn all of the things that we're doing with this kind of research to gather key insights so that we can actually maybe potentially also learn a little bit more about how we can get closer to a vaccine in the future as well. And then the other things is that we know that if we harness the immune system or immune control, that these can have other benefits. For example, there's still a lot of unmet needs and burdens for people living with HIV. We want to try to continuously increase quality of life. We want to continuously decrease stigma that's associated with living with HIV. But also, we also want to try to decrease costs as well, which is would be great in general for anything that we do within the healthcare system. And then when we look at what an ideal regimen would be, we have some considerations that are really, really important. First and foremost, that regimen needs to be very, very safe. So we need to have it at least as safe as antiretroviral therapy. And in fact, we would love that to be even safer or better if we could. And then we also want to make sure that we want individuals to achieve very long viral suppressions. So it's very durable, that response rate. So they can stay off of their antiretroviral therapy for a very long time until such time we can get to cure. And then the other big thing is that we also want to eliminate transmission and, if possible in the future, also protect for against reinfection as well. And then last but not least, and this is the biggest one, is any new regimens that we have that are approved, we really want to make sure that they're affordable for all people living with HIV and that everybody can access it. So we'll go to the next slide. And so you've seen sort of these already earlier today, so I won't talk too much about it. And I think Michael Plus also mentioned about that having no biomarkers. So we have to do this analytical treatment interruption so that we can see all of the dynamics of the viral load. And what's important is that we can also see, okay, when somebody does actually is able to take these sort of immune-mediated regimens and control the virus, how do they compare with those participants that are in the clinical trials that weren't able to control the virus for some whatever reason? So really trying to understand what's happening, because everybody's individual, so we really need to understand everyone. And then the big thing is long-term control. What do we want to do? We said our North Star is cure. We really want it to look very similar to what we see in those post-treatment controllers or those natural or elite controllers that we see. And so a lot of this is sort of regulated by the immune system, and the T cells are really very important for them. We'll go to the next slide. So another similar slide that's maybe done a little bit different way. The big thing is there's a lot of research that's going on in cure, and there's a lot of many different targets, and we're going to today talk about two of them. One is immune checkpoint blockade. So things like anti-PD-1s, and we've heard of that before, very much used in oncology. And then the other one is called immune activation. So regimens that we can actually help the immune system recognize HIV and then elicit an immune response or gain an immune response into it. So if we go to the next slide. So one of the ones that we wanted to talk about today is something called the protein on the surface of immune cells called PD-1 or programmed cell death 1. So essentially what we see is that some of the immune cells like CD4 and CD8 T cells actually have upregulation of this PD-1 protein in HIV, and this may lead to progression. And so one of the big things is that with PD-1, it actually attaches to a co-receptor called PD-L1. And when they are attached together, you have this sort of immune dysfunction, and the T cells are exhausting, and so they can't do anything. So if we can come in with a monoclonal antibody that will block the interaction of the T cell along with the HIV cell, then we can actually have the CD8 T cells release what we call cytotoxic, chemical proteins, and then they can actually kill the virus. So there have been reports in primates and also early reports in people living with HIV that are stable on antiretroviral therapy where we've seen some immune control. But the big question is can we make that durable and a long response? So we'll go to the next slide.
Oh, sorry.
Sorry. So the next target is called alpha-4 beta-7. Alpha-4 beta-7, you see anti-alpha-4 beta-7 monoclonal antibodies in some of our autoimmune diseases like GI diseases like Crohn's disease and those kinds of things. And actually alpha-4 beta-7 could have two mechanisms of action. One of them is actually called FC independent, and one of them is called FC dependent. What is important is these two mechanisms of action do different things. So when we look at it, we can actually sort of stop the transmission, cell-to-cell transmission, if we block alpha-4 beta-7 ligand. The other thing is we can also have it be recognized by immune system cells called antigen-presenting cells. So the alpha-4 beta-7, when it's blocked, it can attach to receptors. They're called Fc receptors, but they attach to receptors on these immune cells called antigen-presenting cells. And what they do is they actually recognize sort of and package the proteins of the virus and put it on the surface of the antigen-presenting cell. So if the T cell comes along, and then the T cell can recognize that there's this virus, and they can actually mount an immune response. And so if we go to the next slide, so essentially, if we could take two of these monoclonal antibodies, when one is an anti-PD-1 and one is an anti-alpha-4 beta-7, the question is, can these have synergistic effects so that we can have a viral control off of antiretroviral therapy, and can that response be very durable? And so what I'm going to do is I'm going to pass it on to Anna, and Anna's going to talk a little bit about our clinical development. Thank you.
Hi, thanks for having us. It's really a pleasure to be here. So Arlene already explained and give you a little bit of an idea of what we are researching. The idea here is really that the anti-PD-1 can make your body fight against HIV again, right? Because sometimes it can be kind of reactivate the immune system, and the alpha-4 beta-7 really focus on your immune system, the person's immune system against the virus. And then maybe this combination will work, right? And we are testing that. And this comes from knowledge from preclinical, right? From another studies that included animals and preclinical. But here, what we are thinking is we give something that will stimulate the person's immune system to fight, and that the immune system is going to take over, right? So it's not something like an antiretroviral that it needs to be in the body all the time to fight the virus. So it's a little bit another concept. And if you can go to the next slide, please. So I'm going to give you an overview of the program. We started with phase 1. This is the first study that we do in people, right? And for the anti-PD-1, we already have, so this is a compound, several anti-PD-1s are being studied in several oncology indications. There are a lot of overlap from immuno-oncology to immune disease and to how the body fights the HIV virus. So we had already data on people coming from oncology. For the alpha-4 beta-7, this was the first time we were doing study people. So you start with healthy volunteers to see if it's safe and if it's okay and how is the pharmacokinetics, and then you move to people living with HIV. So if you can go to the next slide. So this is what we did with the alpha-4 beta-7. We did first the study in healthy volunteers with one dose. Then we look at the safety, pharmacokinetics, and we chose a couple of doses to study people living with HIV. This is study phase 1. Is what we call in phase 1 is for safety, pharmacokinetics. So those people were on antiretroviral therapy. ATI was not done at this point. Okay, and the results of these two studies will be presented at CROI for the first time on March 11th, right? In a poster, and I'm happy to. For those who will be there, we're going to be there to respond to any questions. But we did saw that there was no concern in terms of safety, and there is a good pharmacokinetics and saturation of the receptor. So there is an engagement between the alpha-4 beta-7 receptor and the antibody. This is what we saw in the first. Next one. For the anti-PD-1, we also did one study to understand safety and pharmacokinetics in people living with HIV, single dose first. But then we did a study also doing ATI to see how with the ATI, we would see viral rebound, control, and also the safety and the pharmacokinetics. In phase one, those viral control and are always exploratory. Those are not big studies, right? So there is a lot of limitation. However, which is important to bring to you is anti-PD-1s are very effective drugs in oncology, which is a completely different population, right? In terms of age, in terms of risk benefit, in terms of what you were treating. And they have one important side effect, which is called immune-related adverse event. So as I did explain to you, our body starts to fight stronger and can provoke autoimmune diseases. And that's why the anti-PD-1 block that, right? Because you are fighting, it's not working, so they block and say no more. So when you unblock, when you block the PD-1 and unblock this system, you can have autoimmune disease. And then this is what we call immune-related adverse events. In oncology, you need you need the antibody to block to target the anti-PD-1 in the immune cells, but also in the tumor. So tumors also have this receptor. And the dose that is used in oncology for this drug that we are using is around 250 to 500 milligrams each 2 weeks. And this is done until while it's responding, they continue the treatment. We looked at this and based on the studies that were done in preclinical, we thought maybe we don't need that much. Maybe we just need to block a bit for a limited treatment duration, so it's kind of a weight immune system, and then this may be enough to start a control. And with the other drug that focus the response, maybe you can control and trying to decrease the risk of the adverse events. Okay, so the dose we are using to try to decrease this risk is much less, and we try here from 2 milligrams to 20 milligrams. And what we are using now, and you're going to see the results of the phase 1 study, is 10 milligrams. Okay? Trying to really do appropriate it to the population that we are studying with our people living with HIV. Next one, please. So this was presented in 2023 and also last year at CROI. I'm going to go over quickly, otherwise I don't have enough time to go to the phase 2. But this is the study that did ATI. Okay, the only study in phase 1 that we use ATI. Next one, please. So as I said, Barigalimab, which is the anti-PD-1 we are studying, is a drug used in cancer. Our goal here with this program was, can we use less and mitigate the safety risk and make this work for people living with HIV, for viral control. But of course, the main objective of the phase 1 study was the safety part. And you can go to the next slide, please. Mm-hmm. So this is the study design. The group that I'm going to focus the results is this one. A group of the 2 milligrams we concluded was too low. And this group, what we did is we did 2 doses and then we stopped the antiretroviral therapy after start. And we did nothing more. In this group, we did 4 doses because we already had results from that and we think it was safe enough. But we did start ATI here because you need to make the immune system start to react before you have the rebound. And when you take out the antiretroviral, the rebound is happens usually between 2 and 3 weeks. Because we do think that you need the antigen, you need the virus for the immune system to react. And that's how we build the phase 1. Next one. This is the participant characteristics of the 2 studies that we did with barigalimab, the 2 stages. As you can see, it was mainly white male. We had a good portion of Hispanics and age more or less between 42, 49. with 9 to 19 years, living with HIV, CD4 around 700, and vital suppression on ART for about 10 to 16 years. Next one. Those are the combined data for the two studies in terms of adverse events. So we did saw 3 immune-related adverse events. In oncology, they are very common, like 50%, and the severe ones around 15 to 30, depending on the tumor and on the drug. You can go. Yeah, that's it. Thank you. Next one. Yeah, this one. So we saw 3 cases. 2 of them were alteration of TSH. So one of the things that can happen with anti-PD-1s, they can give hyperthyroidism. Your thyroid starts to work not well, right? And we do have 2 cases that were subclinical, no symptoms, just the lab, and they revert. And as soon as the dose was done, it came back normally, right? Which in oncology we don't see because they keep dosing the person if it's working. And we saw also one hash that can be considered also immune-related adverse event. But we haven't saw anything of the severe adverse events. Those are grade 1, grade 2. We didn't have any colitis, pneumonitis, autoimmune hepatitis, those that we see in oncology. We haven't seen anything in this study. No death, no serious adverse event. This study was limited, of course. It's a total of 73 participants, but 18 to placebo. It was a placebo-controlled study. So there is a limit, very important. Next one. As I said, looking at the viral load kinetics was exploratory. So it was exploratory objectives. However, in this second group that I said we were going to focus, we do see on people who receive the drug that we see as it has explained in the morning, right? The normal one, which is like the placebo effect, that the drug, the viral rebound is very high and very quick, between 2 and 3 weeks. But we see some participants that had some control. And because this is a safety study, of course, at week 12, we said everybody goes back on antiretroviral. But we had 2 participants, and if you can go click for the next, that didn't wanted to go to antiretroviral. They didn't wanted to restart because they control below 100. You can move, please. The slide, yeah. And then they didn't wanted to go back, and they did control for at least the end of the study, and we know outside of the study a little bit more, right? So, and we also see some delay viral rebound, and we haven't seen any of those in the placebo. There is limitation in this interpretation, right? I mean, this is exploratory endpoint, a small study, but we thought that this would, make us very, I would say, excited to move forward with a combination between this drug and the anti-alpha-4 beta-7 in a proof of concept study, designed for efficacy and to see if it's working or not. And this is what moved us to go to phase 2. So if you can go to the next slide, next one. So then, after completing phase 1, as I said, this study was the only one with ATI, the others not. We moved to phase 2, combining both and bringing the doses that we found in phase 1 that were safe enough for participants. We started in 2023. The first site was activated in October. The first participant screen was in October as well. First participant first dose in November. And we just had the last participant last dose in February this year. It's a total of 160 people. And I will show you, and our final analysis is going to be around 2027 because we are looking at 2-year period. Next one, please. So we had we saw that in phase 1 that it was done only in the U.S. That the diversity was, it was more white male. So for the phase 2, First, we really expand to do a multicentric study in several countries, and this includes United States, Puerto Rico, Brazil, Canada, UK, Spain, South Africa, France, Germany, Italy, Belgium, Poland, Japan. And we did select the sites that had first experience with analytical treatment interruption, diversity, staff experience to support the participants during the study. So everything that is said here, we also learned in phase one that it was emotionally very hard for a participant, maybe even if they were like okay with the study and understand everything, when they say they see the Bible going up, it's hard, right? And so we look at for sites that could give this help and this emotional support and really work with them to be able to do a appropriate informed consent, discuss with the participant every detail, explain the risks, right? At this point, we don't know if there are benefits, if it works. We only have a small time, let's say from phase one, right? And of course, the other things is structure for drug infusions to collect PK. This is a very intense study. The participants who join are really like very committed and are heroes here because it's a weekly visit. It's a very hard study. Sites were also encouraged to share and discuss the protocol with their communities, and we got a lot of feedback. I think we rewrite that before the first version like several times. Some things that we learned from you. I mean, we asked it for the sites to every visit discuss with the participant HIV transmission, right? We offer PrEP for the partners, but we didn't enforce anything in terms of disclosure. We just make sure that the site can give this support, and if they need, they will have, but it's up to each participant. But we did reinforce with the site that they need to make sure that the participant, understood correctly that during this time that the rebound is going to happen, there is risk of transmission, right? And they need to be aware of that. This is the study design, so it does include placebo. One dose of the anti-PD-1 alone, you receive placebo for the other one is double blind, right? But monotherapy of the alpha 4 beta 7, and this is a regulatory requirement because you need to be able to demonstrate that the combination is better than the drug alone. And then two combinations with two different doses. And the way it's done, all the drugs are infusions. This purple one is the anti-PD-1, and it's an infusion given on day 1, week 2, week 4, week 6. And the more blue one is the anti-alpha 4 beta 7, given on day 1, week 4, and week 8. So at day 1, week 4, they receive both. First the purple and then the blue. So it's very intense drug, right? And then on day 1, we stop the antiretroviral therapy. It's weekly visits until week 12. And then, depending on how it goes, it can be each 2 weeks. We discussed several times, including with physicians, patients, and to give feedback on how to decrease the burden of the number of visits. And what we end up doing is starting on the week 11, the participant can have a virtual visit that can be either a nurse going at their house if they want and collecting blood and doing the virtual visits, or a virtual visit with the physician and collecting laboratory in any other laboratory that is local, right? And this is the choice of the participant. They can also go to the site, and some people prefer to go to the site, and it's their choice. But it has to be agreed with the physician and the site to make sure it's safe. Right? So we try to build a kind of a flexibility that they can choose, but with safety. And the participants who go back on antiretroviral therapy, they are not taking out the study. They continue in the study. They have less visits when they are suppressed. but they are also follow up for safety until the end of the study if they agree. Next one. This is a bit of the inclusion criteria. So our phase 1 included adults from 18 to 60 years old. Then we had several discussions about feedback. So for this trial, we increased for 70. However, we did, it's a phase 2, so in condition of good health. So they could have comorbidities, but it has to be stable, right? It can be a person that has diabetes, hypertension, but it has to be stable. Antiretroviral therapy for at least 12 weeks, 12 months, sorry, with negative anti-H, HIV RNA undetectable, but we did allow one blip, and CD4 above 500. We kept the nadir below 200 when we wrote this protocol. Exclusion criteria was previous exposure to long-acting, because if you take it out, the antiretroviral, it can lead to, it can get resistance, nadir CD4 below 200, and any medical disorder that, the physician could consider that could, be not safe. Next one. This is measuring safety, and we are measuring not only the adverse events that are related to the drugs, that could be related to the drugs, we are measuring the adverse events that could be related to the ATI. We have a special page to collect RNS or any symptoms that could be associated with viremia. It's also looking at efficacy, which is less than 1,000 copies at week 24, because it takes some time. We learned from phase 1 to the viral load to rebound and then control without antiretroviral therapy. It takes some time. So we have to allow this. But you're going to take a look on the ARCH criteria. And we are measuring the peak viral load and the time to rebound. Next one. So these are the ARCH criteria. So any confirmed viral load above 100,000, if it's confirmed, they restart. Above 10,000 if it rests for more than 4 weeks, and above 1,000 if it rests for more than 6 weeks. CD4 below 350, and in pregnancy, of course, retroviral rebound syndrome. And then we put 2 other criteria that are very flexible, considering that this is an ATI study, which is phase 2. The first one is any adverse event, which in the judgment of the investigator can, propose any risk. Of course, that there are things that maybe we didn't, we're not expecting. And the 8th one is any social or behavior condition which in the judgment of the investigator could pose a substantial risk for the participant or participant request. So if the participant at any time is very concerned and wants to restart, can restart. We don't take out of the study. We keep, monitoring. We're going to space also the visits when they are back to work and resuppress, but we're going to keep monitoring and we're going to look at all the biomarkers, reservoir, everything also on those participants.
Are they told their results of their viral load?
Yeah. So this was also a point of discussion on the phase 1, right? But there is no way you can blind that because it's very risky. Yeah. Next one, we also measure CD4, but we are measuring CD4 every week together with HIV RNA. And then when they are resuppressed, then it spaces a little bit, but it is very frequent. And then we are waiting for this data. As I said, the study is ongoing. It's fully enrolled, but we still have a period of observation. If this shows positive results, then we're gonna discuss the benefit risk of moving that to a phase 3, right? And then to look at other populations that could benefit. We are excluding the people who were treated very early, as we saw the presentation, right, from South Africa. And because we wanted to focus on the chronic people living with HIV because we think right now it's the biggest unmet need. But of course, if you work in this population, we can now look at others as well. And that last slide, this study could only be done thanks to the participants and their families. We are really impressed for the engagement. People who participate in this trial, they are, as I said, heroes. It's a very tough trial to participate. And we do have the people that are super engaged, and it has been really an amazing experience in the engagement. And of course, the HIV community and the study sites as well. Go ahead.
Two things real quick. What are TR adverse events? Who's on the slide?
Treatment-related adverse events. Sorry for that.
The
Yeah.
other thing is just a comment. Whenever I see a presentation where it says healthy adults, it really bothers me. And referring to people who are not living with HIV, who are not HIV infected, who are HIV negative, those people can have all kinds of health issues. They're usually students at university, but they still might drink too much. They may have controlled diabetes. They may beat up their wife. I don't know. You know what I mean? There's so many things that they could have lipids, high cholesterol and triglycerides and whatever. It just bothers me. And I just request everybody just to not do that if they can. And all you can say is people not living with HIV or HIV negative or HIV negative or
Okay. Yes.
Other question, there's been a lot of talk today about the ATIs and follow-up. So in your studies, are you doing any follow-up of the patients long-term after the ATIs to see if there are any adverse?
So we extend the follow-up for 2 years. And if, depending on what we see in terms of results, we will follow for more time or retweet if it fails and if it works and if it fails. So this is under discussion. That's why we did for 2 years by now.
And then what kind of follow-up are you doing? One of the concerns is the neurological complications as a result of, so are you doing any baseline neurologic testing and following up with that?
So we are doing clinical follow-up for neurological. We are not collecting any specific tests. We are on the hope, of course, right now I cannot say we need data to say if it works or not and how much, but we are in the hope that because you are provoking the immune system to control the virus, that this control will go in all, right? I mean, this is the hypothesis, right? But we are following up also with biomarkers, reservoir, immune, several tests to look at CD4, CD8, and intravRNA and CD4 as well.
Just maybe doing something really simple to achieve, like a mocha or something like that. Donald Trump can pass it. Person, woman, man, cancer behavior.
What was I'm sorry, I may have seen at the age. What was the age range?
18 to 17.
Okay, and have you looked at younger versus older response?
Not yet. It's ongoing, it's still blinded. So yeah, we don't have any results.
One of the other big things in ATI studies is the frequent viral load monitoring and developing other points of care, other assays that allow something to be done within the home setting and discussing that. And we've been in there was a lot of discussion at the last Munich meeting about in the ATIs with all of the patients who were cured, all the cure subjects. It was a whole documentary Munich. And we've been actually talking to Abbott about this in the past, about doing this with Gavin. And we haven't really come back to it. So I'm wondering, have you been discussing that across the parking lot?
No, we are discussing because we think it was going to be very helpful, right? For this study, we couldn't do that, so that we build this flexibility of doing in any lab, not certified lab, of course.
You just have to walk across your parking lot.
Yeah. What we so is the nurse. So the nurse visit that goes to the house and collect everything. We put for all the countries available, but there is almost no visit. So most participants they want to come to the site or to do in a local lab and have a virtual visit with the physician. Thank you very much.
Thank you so much. I don't know, can you hear me?
No,
It's different.
can you hear me? I have a question though for ATI, for the field in general. Where are we with alternative biomarkers so that people don't have to actually stop ART and you can monitor? Is that still being pursued or where are we with them?
I mean, in our side it is, but for a biomarker that can mark response versus non-response, you need a study with a good number of people with response, right? With a clear response. Then you try to find if there is a marker there. So it's not that easy. But I think I've seen several AGI studies, not only one, several others that we are looking at this. I don't think right now we have something that we can rely to say.
Because it's so intense for people and the fear of your long-term effects many years down the line.